This lecture covers chapter 15, gene cloning and DNA analysis in agriculture. So far we have mostly talked about how we can modify genetically different kinds of organisms. This chapter is more about why all the commercial strategies that where genetic engineering has been used as a part of that strategy for commercial ends in agriculture. There is a table in the book listing many kinds of different examples of commercial genetic engineering of plants. I chose to talk about 4 of these mainly. Bt crops, that are crops that express toxin that kills insects. Virus-resistant papaya, which is an example of engineering a plant with virus resistance. Roundup ready crops, that is an example of engineering plants to be resistant to certain herbicides. And finally the flavor Savr tomato, which is an example of engineering for quality. So all of these examples cover a quite wide range of goals of genetic engineering. Certain soil-dwelling bacteria, such as Bacillus thuringiensis, have evolved different kinds of toxins, and these toxins are protein toxins, so they are encoded by a gene. And these toxins are very effective against insects. And Bt crops are simply crops where you have taken the gene encoding these toxins and inserted them in plants. One class of toxins produced by Bacillus thuringiensis is called delta endotoxin. And this was one of the first toxins that were attempted to express in plants. The Bacillus thuringiensis Cry1A version B gene, encoding quite large toxin, 1155 amino acids, was identified, and further analysis revealed that the toxin is actually only a part of the protein, the first part here. So rather than cutting the gene up in 2 pieces or using the whole entire gene, an artificial gene was designed for the first part of the protein. This is to also solve the codon bias problem that might occur. This artificial gene was put between a promoter and a terminator that is suitable for the plant, and initial PCR, analytical PCR testing showed that the gene was in fact inserted into the genome. A problem that the scientists working on this project found is that different plants, different transformants varied enormously in the amount of toxin that they produced. And this is a general problem for genetic engineering in plants in general. We believe that this is due to positional effects, that depending on where the gene is actually integrated on the chromosome, or which chromosome the gene ends up, the expression level will be affected by the environment on the chromosome. This toxin here is just one of many different toxins, and the one advantage of the delta endotoxins is that they are quite specific. So there is basically a toxin for every kind of insects. This is good for ecology, since you don't kill the insects that are actually good for the ecosystem. Expression of the delta endotoxins in plants have proven to generate very toxic plants for the insects, which is good, but it also poses a problem, Since insects are quite numerous in number and they are with a short generation span, so it's very difficult to avoid resistance due to natural selection. There are a couple of this was an anticipated problem, and there are a handful of strategies that either have been tested or been suggested to avoid this. The first example here is simply expressing two unrelated toxins. So the idea here is that it's unlikely that one insect is mutated in such a way that it will be resistant to two different toxins. The important here is that these should have some kind of mechanistic difference between them. The other strategy here is simply reducing the exposure of the insects to the toxin by regulating the production of toxins so that it only creates resistant plants when it is needed. So in this example here, it's evidently that you can accept some damage to the plant early in the life cycle of the plant. It doesn't kill the plants if some insects eat of it, but when the plant is ready to make corn, in this case, it also starts to produce toxins, and the plant is resistant and survives and can produce corn in this case. This is not likely to completely abolish resistance among insects, but at least reduce the exposure. It's the same idea behind the idea that you shouldn't overuse antibiotics in society. The third idea is mixing resistant or protected plants and normal plants, these yellow ones here. What happens here is that there should be a certain percentage of these plants in the plantation that insects will both try to eat these, but they will prefer the non-toxic plants, and the majority of population of insects will survive on these plants, which means that it's difficult for a resistant gene, a resistant mutant, to establish in the population, since there are so many non-resistant insects in the population. The fourth possibility is also related to the first one, simply increasing the production of the protein. The book speaks about expressing the endotoxin in chloroplasts instead of in the nucleus, and there are many more chloroplasts than nuclei in the cell, so it was observed that you get a much higher production of the toxin, and this could help help to kill instead of to completely kill off the insects and not establish any kind of partially resistant mutants that can then go on to mutate further and then establish resistant insects. If we look at the third option in more detail, the way this is supposed to work is that you divide your area that you grow your crops into a larger one, 80% or so, where you grow BT corn in this case, and there will always be some resistant insects that develop in this area. But on the side you have non-BT crops, corn in this case, where you have a population of the same insect, but sensitive, because here no resistance is likely to evolve. This means that on overall there will be a much larger population of the sensitive insects compared to the resistance, and by classical population genetics the resistant genotype will have a different or difficult time to establish itself in the population, because it would be dominated by the sensitive alleles. In order to solve the problem, or the potential problem of glyphosate accumulation in the plant tissue, another kind of resistant mechanism was employed. A gene called GLIAT that encodes glyphosate and acyltransferase was found in a bacteria Bacillus species. This enzyme can attach a acetyl group on top of the glyphosate molecule. This inactivates glyphosate and it also directs its degradation. This could perhaps be an alternative mode of resistance, so that the added herbicide is actually degraded in the plant tissue so that it doesn't spread in the environment. A problem here was that the enzyme was not very active, but a very interesting technique was used in order to isolate highly active GLIAT enzymes. Three different genes of the GLIAT, three different GLIAT genes from different organisms were cut into pieces and shuffled together by PCR. So they are so homologous or so similar that they can anneal with each other, fragments, and the gene shuffling increases the diversity between the three. And from these diverse pool of new GLIAT genes, one that was thousands of times more active was isolated. This is a very powerful method, and it's been used in other situations as well. This enzyme was able to create glyphosate resistant plants that could tolerate high concentrations of glyphosate, 6 times higher than necessary. I'm not sure that this has been commercialized yet, but it's probably in the future. The delta endotoxin is produced in the form of crystals during the sporulation of the bacteria. You can see here a spore with a kind of diamond-shaped crystal of the crystallized protein, really. And these toxins are, as I said before, quite specific, and they're active against several families of insects. The protein itself, the endotoxin, is quite interesting mechanistically. It has these 3 domains: the blue one, red one, and the green one. Domain 1, 2, and 3. And the first domain is involved in a process called pore formation. The red domain 2 here is proteolysis and specificity, and the domain 3 is involved in receptor binding. And all these domains are important for the function of the toxin. The crytoxin has a very interesting mode of action. During sporulation of the bacteria in the natural source of the toxin, it crystallizes in these diamond-shaped crystals here. And when these bacteria are ingested by the insect, the environment in the gut of the insect solubilizes this crystal, Insects have high pH in their guts, as opposed to mammals like humans. We have low pH. So the high pH favors the solubilization of the toxin proteins. Once they are solubilized, they are processed proteolytically. So this gray domain here represents the C and the N terminal of the protein, and specific proteases cut these away from the protein. The toxin has 3 domains. This red one here, it's domain 3. This blue one is domain 1, and this green one is domain 2. And they all have their specific role in the action of the toxin. The domain 2, the green one, binds to specific receptors in the gut epithelial cells of the insect, so inside the stomach of the insect. And when that binding occurs, this induces a conformational change, mainly in the domain 3, the red one here. And when that occurs, this structure can pair with other identical structures, and this red part, the domain 3, forms a pore. So here we have the red part represents the domain 3 here, and the green and the blue the other domains, and they pair together or with many copies of itself, and they form a pore through the epithelial gut cell wall of the cell. This means that water will escape and the insect will dry out and it cannot feed anymore. This slide here shows a couple of images on what happens when insects ingest the Bacillus thuringiensis toxin. As you can see here, this might be the normal, the way the insect look normally, and then after feeding for a short time it becomes all dry and black. You can see some examples here. Research has been performed in order to find possible mechanisms of resistance to these toxins. This slide here shows a picture of a nematode. It's a C. elegans, which is, there are toxins that are that will attack this organism, and the research was performed using fluorescently labeled toxin. So you can see here that in a normal worm the toxin seems to attach, it lights up the inside of the worm. But here we have a mutant that is somehow mutated so that the toxin doesn't seem to bind to the interior of the worm. And this was not something that was found in nature, but a mutant that was isolated in the laboratory in order to study possible mechanisms of resistance. In this case, the enzyme or the gene that was changed is called BRE, and this is a gene that encodes galactosyltransferase in the nematode. And this is an enzyme that adds carbohydrates to proteins and lipids. And this has an effect because what is actually recognized by the toxin here in red, and we see the molecule here, this molecule is bound to it's a sugar polymer, and it's bound to the membrane protein to which the toxin binds. And if this galactosyltransferase is not active, then these chains of sugars are not transferred to the membrane protein, and the toxin will not bind. So this is one possible mechanism by which resistance could develop. The diamondback moth is an insect that causes lots of damage to plants such as cabbage and kale. And this was the first insect that developed resistance to the Bt toxin in nature. This is a very old study published in Nature in 2003, almost 20 years old. And it describes resistant insects in the 1980s and 90s, so that these resistant insects arose from the use of Bt spray, that is Bt toxin isolated from bacteria and then applied on top of the plants, so it's not recombinant plants. As far as I could see, there is no examples of Bt resistance developed from transgenic crops. Typically, the dosage of toxin in transgenic crops is far higher than than you can apply on top of the leaves with a spray. There is another insect as well that has been described lately to show resistance to the Bt toxin. One reason why this particular insect could have acquired resistance is that it has a very short life cycle, so 14 days. So this means that beneficial mutations can arise and establish quickly in the population. I will now switch focus to Roundup ready crops, and I will start talking about herbicides, different kinds of herbicides. There are selective and non-selective herbicides. The selective ones kill certain types of plants, but now not others, depending on biochemical differences in their metabolism. As you know, we divide plants into crops that we want and weeds that we don't want. And there are many examples of selective herbicides that will kill weeds but not crops, depending on what actual plants we are using. Then there are non-selective herbicides such as Glyphosate or Roundup. This is sold under the commercial name of Roundup. This is a very simple molecule. It's a glycine amino acid with some substitute groups here. And this is a non-selective, so it will kill all plants basically. This makes it less useful because you have to be careful on how you apply it, because otherwise you kill your crop as well, in addition to the weed. The glyphosate molecule inhibits an enzyme called EPS-PS synthase. So this is a enzyme in the Shikimate pathway. So this pathway creates intermediates for different kinds of amino acids in the metabolism. These amino acids are necessary because plants synthesize all their amino acids. So if we inhibit this enzyme, the plant will die due to starvation of these amino acids. Glyphosate is a competitive inhibitor of the enzyme, and it competes with phosphoenol pyruvate in the synthesis here of enol pyruvyl shikimate 3-phosphate. It was later established that if there is a mutation in EPS-PS synthase, if we switch, there is a possible mutation. We switch a codon so that the proline in position 101 becomes serine, shown here. Then glyphosate no longer binds to the enzyme, and the enzyme can function and produce its product, and hence all the amino acids, even in the presence of glyphosate. This means that we can genetically engineer plants with this modified enzyme and then grow and spray this Roundup that normally would kill plants, and we kill all regular plants, weeds, but our Roundup ready crop survives. This works all very well. One disadvantage perhaps is that this kind of resistance doesn't inactivate the herbicide. So this enzyme is simply insensitive to the herbicide, but there is no process by which the herbicide is deactivated, which maybe would be advantageous so that the Roundup doesn't accumulate. Now I will switch focus again to the flavor Savr tomato that was genetically modified to increase its quality. Before I go into that, we have to talk about how tomatoes ripen. What is the ripening process of tomatoes? Tomatoes have pectin, a sugar polymer, in their cell walls, and the pectin gives rigidity to the cell wall. So there's lots of this pectin in an unripe tomato, in a green tomato, for instance. What happens, among other things, during the ripening process is that a polygalactoronase enzyme is expressed, and this enzyme softens the cell walls by degrading the pectin in the cell wall. And this is desirable, but a problem as well, because this makes it difficult to handle tomatoes. Ripe tomatoes are almost impossible to transport, for instance, and to handle in a shop. So that's why shops usually buy green or unripe tomato and let them ripe inside the shop. This is not as nice as sun-ripe tomato though, so that's a problem. It would be nice if one could handle ripe tomatoes in a practical way. So the scientists set out to solve this problem. The scientists already knew that what the polygalactoronase gene was, and they set out to delete it, to silence it. This was done many years ago, so direct deletion of the gene was a much harder task than it is today. What the scientists did was to design an antisense gene that would express and silence the gene. That is a gene that is complementary to the gene itself. The way they did this was to isolate the front part of the gene and put it in reverse between a promoter and a terminator in the plant, so that the normal direction of transcription is this one, and then the gene is transcribed in reverse in this case. So how does that work in detail? If we look at this antisense technology in more detail, we have the normal gene here, that is in the double-stranded DNA here. We have a start and a stop, and the transcription results in a normal mRNA. Normally this will then go on to be translated by ribosome. But in this case we also have an antisense gene, so it's the opposite gene, in the opposite direction, like in the slide before. We get also an mRNA here. This one cannot encode a protein. Of course it would be clearly an inactive protein, but that's also not the point. These two molecules are complementary to each other, and if they combine this way they will not be translated, but they will be degraded, and in this case silencing of the gene, that there is no expression of this gene. This is an advantage of this technology. If you have many copies, for instance, of the gene, instead of deleting every gene, you can simply express at a high level the antisense gene and silence all of them, for instance. In this case, the reason was probably different. This was done before there were even PCR, so in terms of technology, it was quite a feat. This gene silencing was proven to work. You can see here the control, unripe tomato, and a ripe red nice tomato here, and this is the control, so this is the normal tomato. We have the antisense expressing tomato, starts off almost exactly like the control tomato, but after many more days, almost twice the time, the tomato is still not squishy, so it will easier to handle, but it has many of the flavor ingredients that the ripe tomato has. And they also could measure the activity of the enzyme and notice that the transformed plant continued on a low enzyme level. Although this, from a biological and biotechnological perspective, worked, as a commercial product, the Flavor Savr tomato failed. And it failed for two reasons, mainly. This was commercialized in 1996 around in the UK, but EU law made it mandatory to add this label to the tomato can, made with genetically modified tomatoes. And that wasn't very appealing to the consumers. Around the same time, competing tomato called the LSL tomato with similar characteristics was also marketed, and this one was made with classical genetics, so you didn't need to add this label to the cans. Another problem was also that Calgene, that was the company that developed the tomato, lacked experience in growing tomatoes, so these tomatoes were more durable than normal ripe tomatoes, but not as durable as green tomatoes. So lots of tomatoes were lost initially in the process until they got that right. So all of this made it economically inviable to market these tomatoes. But it remains the first example of many different gene subtraction projects that are listed in the book in table 15.3, where you modified different kinds of plants in different ways. I will now switch focus again to the development of transgenic papaya fruit that were resistant to ring spot virus. Papaya is a big commercially important product in Hawaii, and what happened in Hawaii in the 1940s was that the Second World War brought lots of people and other things to the islands, and among the things that they brought was the ring spot virus that affects the papaya plant. You can see here the difference. This is a normal papaya plant, and this is one affected by the ring spot virus. You can see the spots here on the leaf. This plant will not produce any papaya at all, or very little of it. And you can see here how bad this is for the plantation. This is a big economical problem because papaya was a huge export for these islands. something called coat protein mediated resistance was employed in order to create resistance to this virus. This is the normal papaya fruit, by the way, and this is a fruit. Here we can see the ring spots. They look like rings on the surface of the papaya. What was done was simply to express protein, a coat protein from the virus. So virus genomes usually contain a small number of genes, usually RNA polymerases, or but some of the proteins are usually coat proteins. So they encode for proteins that protect the RNA. And it was discovered that if we express this coat protein in the plant, then the virus cannot infect. The precise mechanism is not known, although it has been investigated. Several different mechanisms have been identified, and the most plausible explanation focuses on the mRNA, that somehow the mRNA is somehow the production of mRNA of the virus is affected by already expressing this protein. Another mechanism is the assembly of virus itself. So by already expressing the coat protein of the virus, it is thought that this overexpression of the coat protein, together with the virus's own expression, somehow interferes with the assembly of the virus, so that the virus that are produced cannot infect new cells. Whatever the mechanism, this was quite efficient. You can see here an example of a variety of papaya called Rainbow that is made to be virus resistant. Here you can see the construct that was used. This was done many years ago, so kanamycin resistance was used together with reporter gene, the goose reporter gene, and this construct was linearized and integrated into the genome. You can see here the effectiveness of the plant. You can see here the central plantation of the transgenic plants and the non-transgenic plants outside, and it's quite obvious what the effect is. If this was done many years ago. Today it would typically not include a resistance gene, but you would do a clean genetic engineering instead, because you don't want to spread resistance genes in food. They might, or very little risk perhaps, but they might transfer to your gut biota, and you will have antibiotic resistance genes in your stomach, and that's not good. Living organisms like plants have one thing in common with software, which is that if you have a copy, you can easily make new copies. If you have a plant, you can quite easily make new plants by collecting seeds, and then you only have to buy your seeds once. This is not good for the companies, of course, that developed genetically engineered plants. So they decided to employ a copyright mechanism into their transgenic plants. This is called genetic use restriction technology, or GERT, and this is an outline of how it works. The idea here is that the farmer should buy seeds from the company every year instead of using seeds collected from last year. The way to achieve this is to make sterile plants And how do you make that? First, toxic gene is isolated or invented. This one particular gene is called RIP, ribosome inactivating protein, that inactivates the ribosome and kills the plant. This gene is modified with a blocking DNA inside. So a plant cell is transformed with this gene that has blocking DNA. It's put behind an embryonic promoter. So this is a promoter that is only active in a certain stage of the plant's life cycle, in the embryo life cycle. This is a stage after the unicellular stage, but before the plant actually germinates. This construct it was put inside the cell, and then an embryo was created, or the stage of the embryo was passed. So this is a collection of cells. Then this blocking DNA was flanked by specific sites for a recombinase, CRE or Cre recombinase. This recombinase was added to the cell it was also present but induced by tetracycline. So this is an inducer, the promoter that is sensitive to tetracycline. We can turn on the production of the tetracycline, and then the blocking DNA disappears from the RIP gene. This is okay because this promoter is not active. The stage here, the embryo stage, has passed and the gene is active. We can use this seed to grow a mature plant with no problem. But when this plant creates new seeds, then these seeds will pass the embryonic stage at which the embryo is, this promoter is active, and they will kill, the plant will kill its own seeds. This way you can make sure that the farmer buys seeds from the company every year. It's of course not a popular thing, and I don't think that it was approved ever for commercialization. There is an alternative, more advanced method employed right now, where instead of creating sterile seeds, the seeds that are produced don't have the genetic advantages that the GM crop has. So instead of making it necessary to buy new seeds from the company, you can use the seeds if you want, but they won't be genetically modified. And this is a much safer route, and it makes the farmer less dependent on the company. Genetic engineering can also be used to create new art. This rabbit here was perhaps the first example of transgenic art. So this Brazilian, I think, artist created a transgenic rabbit that expresses GFP, green fluorescent protein. So it has this green shiny light under fluorescent light. He went on to create transgenic dogs and other animals, and this has no purpose, has no practical purpose, but as a art statement. I think this was done very long time ago, 20 years ago, and there are perhaps many different examples of transgenic art nowadays. You can even buy your own transgenic organisms online. There is a company selling transgenic fish that you can buy, and these are zebra fish. So zebrafish is usually used in biological research because they're easy to manipulate. And these 6 zebrafish here each express a different fluorescent protein with different color. So the zebrafish is normally white, has no color at all, but here it expresses different kinds of proteins, and if you have the right lamp in your aquarium, you will create quite a spectacular effect. It is unclear whether this is legal in the European Union. You can buy and sell these fish in the United States. However, analysis or a survey was made in animal shops in Holland, I think. And these fish, although perhaps not entirely legal, were available in many places in these aquarium shops. So perhaps the genetically modified organisms are spreading and we don't really know about it.